Thank you to the organizers for giving me a chance to speak today. So I'm going to tell you about some work that my lab at Einstein has been doing during the pandemic. A lot of the work that I'm going to tell you about is being done by this group of people. That's about half my lab. That's we're informally calling the COVID crew, and I'll call them out as I kind of go through the work. But this is also a much larger collaboration with a number of other labs on campus and off campus on the various projects. And again, I'll try and credit folks as we go along. So the way we got involved in working on COVID was that Lizan Pravsky, who's the chief of clinical ID and a professor at Montefiore here and a professor at Einstein, was the PI of a clinical trial to look at the utility of convalescent plasma. And so she reached out to me to see if we could help to look at the magnitude and the nature of antibody responses in COVID-19 patients and convalescents as part of this convalescent plasma trial. And so she asked if we could identify convalescent plasma donors with high antibody titers to treat patients. And, of course, this leads to a number of research questions that a lot of other folks are also addressing. What are the correlates of protection afforded by CP? And then, something that we're interested in as a lab, which is, can we mine B cells from convalescent donors to find potent monoclonal antibodies for prophylaxis and therapy? And so this is one group of collaborators that's involved in a lot of the work that I'm going to show you that relates to looking at serology in convalescent and patient blood samples. So first we had to find these donors, and this was really a huge effort by Johanna Daly, ID doc at Einstein, professor and a professor at Einstein. And so she really went to the areas that were immediately affected in early March, this huge cluster in New Rochelle, Scarsdale, further north in Rockland County, and then a bit later even in our own hospital, Montefiore Medical Center. And these are just some pictures showing you some of the collections that took place as part of this huge effort. And we received hundreds of blood tubes from these individuals from whom small samples had been collected as a pre-screen to identify people that could potentially give plasma. These are all volunteers who wanted to give plasma for CP therapy, but we wanted to make sure that we actually got people who had good levels of plasma, of antibodies against the spike protein of SARS-2 in their plasma in order to be able to give that as a treatment. So our first task was really to develop a serologic assay for SARS-2 spike-specific antibodies. And just again, I think for the group that's attending this webinar and all the other participants, I probably don't need to go into this very much, but I'll just briefly point out that the spike protein, or the S protein encoded by the S gene in the viral genome, forms this kind of corona on the surface of the virus particle, which gives this whole family of viruses their name. And it's a trimeric spike that's a viral membrane fusion protein that structurally and functionally, at least in broad outline, resembles the glycoproteins of HIV and Ebola and influenza and a number of other so-called class 1 fusion glycoproteins. So the spike protein of SARS-2 mediates viral membrane fusion of the virus particle with the cellular membrane and allows escape of the viral nucleocapsid core into the cytoplasm, where it can reprogram the cell to produce more viral particles. And so if you're this is the major surface exposed protein in the virus. This is the protein that's thought to elicit all of the neutralizing antibodies, for example, as part of the humoral response that bind to the virus and block infection by blocking either binding the virus to the receptor or some step in the entry process, or by targeting cells that are already infected and displaying the spike protein on their surface. So we decided we would use this protein to actually develop a serologic test. And we were greatly aided in our efforts by the development of a stabilized recombinant spike protein trimer by Jason McClellan, one of our collaborators at University of Texas, Austin. Jason sent us the construct, and Jean Lai and Steve Almo's groups at Einstein figured out how to produce this protein at high yields in Chinese hamster ovary cells so that we could actually get 20 or 30 milligrams of this protein per liter. This protein, I'm not going to show you the data, but it's really nice protein. It's a trimer. It's in the prefusion conformation. It's suitable for structural studies. It's very well behaved biophysically. So we felt really confident going in to use this as our sort of antigen to find and capture and detect antibodies against the spike protein in patient samples. And so I'm just showing you here and so we actually set up a very simple ELISA assay where we just capture the spike protein on ELISA plates, and then we use that as baits to fish out the IgG and IgA subtype antibodies in patients' serum or plasma. And this is just showing you some typical data from this spike ELISA. Each of these lines or curves is a different patient serum sample that's been serially diluted. And so you can really see that we see, and these are all people that had positive swab tests by RT-PCR. So these are all individuals that were exposed to SARS-2 that are at least 2 weeks out in terms of symptoms, that were all healthy when they were sampled. And you can see there's actually, and as we're like learning now from a lot of the other work that's going on all over the world, the response, the serologic response, at least in terms of antibodies, is very variable. We have the whole gamut of reactivities, all the way from essentially background to extremely strong reactivities. And one of the things we observed pretty early on with this assay is that it is specific, but also has a really high dynamic range, which is great for detecting this broad sort of range of antibody levels in convalescent and patient samples. So this is just tabulating as a histogram what the spike-specific IgG levels are in these convalescent donors. This is looking in serum from over 200 donors, potential donors. The titer is sort of is determined by looking at these curves. It's a twofold signal over background, and it's plotted as a log3 on a log3 scale, since we did threefold serial dilutions, just easier to plot it that way. And you can see again that there's a huge range of titer. The median is about 7,000, but we also have this really long tail at the bottom of people with pretty low levels of IgG to the spike protein in convalescence. So we actually use this as a screening tool to identify individuals with a reasonable, and of course here we're flying blind a little bit, but we chose a titer of 1,000 as a reasonable cutoff for donor referral, and individuals above that titer at the time they were sampled were referred for plasma donation to the New York Blood Center. We then got tails, or these little segments from the plasma bags, when these plasmas were used to be given to patients. And we titered those as well. And it was reassuring to see that we got very similar reactivity in these plasma segments, except to say that this tail sort of disappeared, which was also consistent with the fact that we sort of used this titer as a filter to identify people. So we didn't see a lot of individuals with very, very low titers. So this was reassuring that what we saw in the plasma reflected what we saw in the serum several weeks prior to that. So a lot of this plasma has actually gone into patients as part of the expanded access compassionate use protocol that was approved for Mayo Clinic and then widely used by many different hospitals. And a lot of this plasma is also being used in an ongoing randomized control trial at Montefiore and NYU Medical Center. And so far, our data look pretty promising in terms of, the plasma does seem to improve outcomes, but it does appear that there is a correlation with the initial level of antibodies in an individual, in a patient that's receiving the plasma, whether the donated plasma actually makes a difference or not. I'm not really showing you any data there because this is ongoing. And we're sort of still working through all of that, but we hope to really be able to use this type of analysis to understand, to develop a metric or some sort of algorithm to decide who would be best, suited or would benefit the most from receiving donor plasma, and which patients would probably do better with some other type of intervention. This assay was really set up as a quantitative ELISA to measure antibody levels in individuals, both donors and patients. But as we developed this ELISA, we began to speak with scientists at the Einstein and Montefiore Pathology department that were really doing clinical diagnostics for COVID. And it seemed to them that leverage this ELISA to develop a diagnostic antibody test. So go from a quantitative test to a single point plus minus test that could be used, at our hospital and our affiliate and our clinical affiliates as an antibody test for COVID-19. And of course, the question is, why would you want to do a laboratory developed test? Why would you want to develop such a test for COVID-19 serology? When we started this work in March, there was really an acute need, as there continues to be, for antibody testing for patients and healthcare workers. There's a shifting landscape of commercial tests. The sensitivity and specificity of these tests is not always clear. Sometimes involves small sample sizes. There's also a continuing concern that supply chain issues would make it difficult to keep running these commercial tests. And so, could we future-proof what our pathology department was doing by developing our own test with off-the-shelf research reagents? Of course, to use this assay clinically, anything we develop in the lab, it would have to be ported to the CLIA lab. And so that was really part of this undertaking. Could we leverage the spike-based research ELISA to develop a single-point qualitative test that could then obtain approval from New York State to run in a CLIA lab for actual patients? So again, this is a huge collaborative effort with a number of individuals at Einstein and Montefiore. And a lot of the work that I'm going to show you, we met every week. Actually, we met every day for a period of time, and now we're meeting weekly as the work is sort of proceeding. We've actually submitted an application to New York State that's under review, so actually the assay can now be deployed in the hospital, and I'm going to show you in a bit. So again, this clinical test really leverages this recombinant spike protein that I told you about earlier. And so our charge here was to take this quantitative test where you run 10 dilutions and turn that into a single dilution test. And so to do that, we actually scouted 3 different dilutions for the IgG, 1 to 100, 1 to 1,000, 1 to 10,000. And the bottom panel, the top panel shows you the original data we obtained from some of the convalescent donors. The bottom panel is the same samples rerun in an independent experiment, but only with the 3 dilutions. So you can sort of see that we get very similar data. And so we started with these 3 dilutions, and then we asked which one would work the best in terms of sensitivity and specificity in our assay. We did something very similar for serum IgA, which we felt could be very useful. We didn't really know how useful it would be from a diagnostic standpoint for convalescents, but we thought that at least for acute patients, it could be useful to actually detect and measure the amount of serum IgA, because there's all this data now out there suggesting that a spike-specific serum IgA may actually be a biomarker for severity of disease. I don't think this is totally figured out, but certainly was something that we thought we should measure. We did a similar analysis here, but with slightly lower dilutions, since the levels of IgA were lower in general in the samples. And then we took advantage of the fact that we had this huge cohort of convalescent donors who had provided serum, and we were able to procure over 250 pre-COVID control samples from the Einstein biorepository from previous projects. And so we essentially used these 2 sample sets to very carefully examine the sensitivity and specificity of the assay at these 3 different serum dilutions. And I'm just summarizing for you the data here. And an important piece was also how reproducible is the assay at these single points. And so this is showing you data at 1 to 1,000 serum. These are, so just to clarify, all of the data I'm showing you were run manually by individuals in my lab. The only automation here is the plate washer and the actual reader. All of the other steps were done using a standard SOP under conditions as well controlled as possible. And each of these data sets, one on the X and one on the Y axis, were done by different individuals, sometimes on the same day, sometimes on different days. So all of this just to make the point that the assay is very robust and reproducible, even at a single point readout, and we expect it to be even more so once it's run robotically, either at Monty or elsewhere. So using all of this sensitivity and specificity data from convalescents and controls, we conducted an ROC analysis that's shown here. And here I'm only showing you data for 1 to 1,000 dilution for IgG and 1 to 200. You can already see from this that the IgG assay has a better performance characteristic than the IgA, which makes sense because it's pretty clear that IgA levels are generally lower and seem to wane a little bit faster than IgG in this convalescent group. This is plotting the same data in a different way, just summing the specificity and sensitivity to maximize that, to pick our cutoffs for deciding what's positive and what's negative in the test. Of course, we're cheating more towards the side of high specificity. And so we sort of chose 99% specificity as our goal and accepted the sensitivity we got at that cutoff, at that benchmark for specificity. And so for the IgG, an absorbance cutoff of 0.9 gave us sensitivity of about 90% in this convalescent donor cohort. And so that's what we went with. And for the IgA, the sensitivity was a little bit lower, well, significantly lower at 70% using the same cohort. And so this is just showing you the same scatter plot data, but this time with the cutoffs overlaid for IgG and IgA. And we use this information then to determine the overall characteristics of the assay, which I'll show you on the next slide. But first, we also wanted to make sure that this assay could differentiate between SARS-2 and other circulating human coronaviruses. And so we actually were able to get samples both from Montefiore and from a collaborator in Sweden, Matthias Forssell, a set of curated serum samples from individuals of pre-COVID samples from individuals who had confirmed positive swab tests by qPCR for different alpha and beta coronaviruses, and those are shown below. So MERS, HKU1 to 99E. So we actually did 4 different ELISAs in these experiments, one against each of the 4 spike proteins at the bottom. And so you can see that a number of these samples are positive for HKU1 and or 299E, as you might expect. These people are several hundred days out from being diagnosed. But of course, this is a recurrent infection in many people. So nevertheless, it was nice to see that we could detect antibodies to these other spike proteins in these samples. However, all of them were negative or or borderline or just below at or below the cutoff for SARS-2. So in general, we're able to distinguish between SARS-2 and other human coronaviruses. And so this is just showing you some of the tabulated data from this analysis. For the IgG, we have a sensitivity of about 90% with specificity of about, sensitivity of about 91% with the specificity of about 99%. And those numbers are 70 and 99% for the IgA assay. One thing we have been doing lately in collaboration with Stephanie Forrest, a clinician at MMC, is to actually do a comparative analysis of our assay versus a number of other assays that are currently in use at Einstein and elsewhere. At Einstein, the Abbott N-based antibody IgG assay is sort of a workhorse. And so we especially compared it to that. And we noticed something interesting. In general, there's very good concordance between our assay and the Abbott and a number of other platforms, including the New York State Wadworth test, which is a Luminex-based assay. I'm not showing you all that data here. But one interesting little vignette is that when we look at Abbott's samples in the Abbott that are just below the Abbott cutoff for calling positive, so these are really, the Abbott assay is a plus minus, so there's no indeterminate, but this is, really at the border, right below the border. It was interesting that in our antibody test, we could actually call a number of these as positive, and those are shown in red, while others remain negative. So we have our own set of indeterminates, but at least and we're trying to expand this analysis to more samples to make sure we're not just sort of looking at a small set here. But it does look like, at least in certain situations, our assay is quite sensitive compared to at least this one very well utilized commercial test that's out there. So just to kind of bring this full circle, we started with a quantitative assay and we wanted to turn it into a qualitative single point assay. But as this pandemic progresses, it's becoming more and more clear that we actually would greatly benefit from having a reliable single point assay that can actually give us some quantitative data on on the levels of antibodies. So, so we asked if we could actually go backwards and close the loop. So what you're looking at here is absorbance at 1 to 1,000, which is sort of the conditions that we're using to call a positive or a negative in our antibody test on the y-axis. And on the x-axis, you're looking at the log of the titer that was actually determined from full ELISA curves. Again, this is for the more than 225 or 230 convalescent serum samples that has really kind of driven a lot of the work that we've been doing. And of course, these experiments are done independently at different times. And you can see that despite that, there's actually a really nice correlation between the absorbance, you know, at 1 to 1,000 and the titer, which means that we can actually fit this to a curve and use it as a standard curve to take a single absorbance measurement with our serum sample at 1 to 1,000 and use that to read out a titer. And that's shown here with another set of samples that weren't part of the training set. And the blue bars are showing you the actual calculated titers from ELISA curves. The salmon bars are showing you the data that are interpolated from our curve fits. And the error bars here are really just the 95% confidence intervals for the values reported from the interpolation. And you can see that it's not perfect, but does this is like brand new data from the last couple of days. We have to do a lot more, but it looks quite reasonable. The other thing we're doing is Gaur Kalasa, who actually was at Columbia before he came to my group, is doing a more rigorous supervised machine learning process with this data set to actually see if we can improve the model and really look at the performance of the model in a more rigorous and quantitative way. So we'll hopefully have that data to present in some forum soon. So where do things stand with this? We've validated this against the New York State test. We've actually been collaborating with Steve Spitalnik, Eldad Hode, and others at Columbia. And we've had these weekly calls for a while, and we've exchanged samples and really tried to validate each other's assays and have managed to do that. We have a larger cross-validation study at Montefiore that's almost complete. And we've also applied for New York State emergency use authorization that's under review. And so the work is ongoing now to clinically translate this assay, and we hope to deploy it very soon. Okay, so I'm actually running out of, I'm not sure how much time I have left. Steve, do you know how much
You're
time?
at the 2-minute, 10-second point.
Okay, so obviously I bargained, I didn't go as fast or probably put too much in. So I'm just gonna use the 2 minutes to just very briefly summarize a second piece that I was hoping to tell you about, which is a BSL-2 assay to actually do virology with the spike protein. For a number of different reasons, you would want a neutralization assay to actually to screen inhibitors and profile the immunologic activity of candidate vaccines. And so our approach to do this was to build a recombinant vesicular stomatitis virus expressing the SARS-2 spike protein. This is showing you Ebola, but it's the same principle, where we can engineer the genome of VSV to express the spike protein that we want, in this case, SARS-2. This virus also makes a reporter. And so we were actually successful in making this virus, although it was quite difficult. One really interesting thing we observed, and you can see that in these bottom immunofluorescence panels, the virus, when it infects cells, the cells fuse together and make these giant syncytia, because we think that the virus is putting tons of spike protein in the cells that are causing cell-cell fusion. You don't see that with the SARS-1 virus. But interestingly, as we passage this virus, this phenotype of syncytia went away, and we got normal-looking plaques by passage 7 or 8, and you can see that in passage 9. The virus also started to grow way better. We started with the 10 to the 3 virus. We're now at 10 to the 8 infectious units per mil with this virus, so we can really produce it in large amounts. So why did the virus change? How did the virus change, and why did that help it grow better and not make syncytia? One key thing is it introduced a stop codon in the spike protein that truncates it and deletes the cytoplasmic tail. And a number of other people who've made this virus, same type of virus, have seen this. And we believe it's just retargeting the spike protein from the Golgi to the cell surface where VSV buds, as opposed to coronaviruses, which bud into the ergic. So this is really a particle biogenesis and assembly compatibility issue that the virus has figured out how to fix for itself. And we've obtained a couple of other mutations that seem to just reduce the fusogenicity of the spike in tissue culture. But importantly, this virus still needs ACE2 because if you don't have ACE2 in cells, you don't get infection. You add in ACE2, you get infection. You can block infection with antibodies against ACE2 or against soluble receptor binding domain of the spike. And we can infect respiratory epithelial cells and block infection in these cells with an antibody against ACE2. So the virus behaves as it should for having the spike protein. The most important thing for purpose of this talk is that we can actually use this VSV now to do neutralization studies with serum and monoclonal antibodies with very good correlation between the VSV on the y-axis versus authentic neutralization on the x-axis. This is with the panel of the same convalescent sera I was showing you earlier. And we're currently actually, I'm just going to stop there, but we're also looking at a huge number of monoclonal antibodies that we can screen with our system to identify candidates that would be useful for therapeutics. And I just wanted to just quickly, let's see, just go to the last slide so I could show you if I can figure out how to advance my slides. Okay. Lastly, I just wanted to thank my lab, especially the COVID team, but also everybody else in my lab that's really contributed to this work and all our collaborators. And this is Alternative Careers Day on Halloween, and you can see we have a lot of bakers and a lot of people who wanted to take up the cloth, which is interesting. I guess science does that to people. So with that, I'll stop since I'm out of time, and I'll take any questions.
Thank you, Kartik. And if you could lower your slides so that we can get the next speaker slides loaded.
Yeah.
I believe we have time for a few questions. If any of the attendees raise their hands, we're able to call on you. If not, I'll start off with a question just asking, so you prefaced the introduction to the development of your assay as a means of sort of overcoming the problems with the commercial assays, which we all see are relatively numerous. On what scale do you envision deployment of these assays in New York, assuming everything goes well?
I mean, the assay is built, the base of the assay is this recombinant spike protein, and we can produce it, quite readily for like thousands and thousands of assays. So we don't think that's really a limiting reagent. And all of the other reagents are sort of off-the-shelf reagents that can be used, obtained pretty readily, but still available. So, I mean, I think scaling is not really an issue. The logistics of scaling is not really the issue. I think it's really more about adoption and who might want to use it. I think there's also as sort of things shift, I think, this idea of using a single point assay to actually quantitate IgG responses in individuals who who might be candidates for a vaccine, who have maybe gotten the vaccine, and looking at their sort of serology over time. An assay like this would be quite useful for that, because a lot of the of course, a lot of different companies as well are developing single point quantitative assays, but currently a lot of the commonly used diagnostic tests are qualitative. So I think one of the nice things about this assay is its ability to be used in a quantitative setting and with a single point measurement, so that you don't have to do a whole bunch of serial dilutions. So we're hoping to sort of develop that further and refine it so that that would sort of provide a piece, a utility piece that that would see it more widely used. Also, I think that because you don't need any sort of proprietary sort of machine, or it can be run in any kind of standard research lab, even in like a teaching lab. All you need is an ELISA plate reader and some basic reagents. It also seems like a type of assay that maybe could be used much more widely on a global level. And we have, we are publishing detailed SOPs that people could, if they follow that, they should really get similar qualitative and quantitative data. The assay has been actually run in multiple labs already. And we know that it's quite portable, and it's also being run robotically at Montefiore and with very similar results.
All right, so we have a question then from David Cohen. Go ahead, David.
Have you looked more specifically at the binding of these antibodies to the where on the spike protein they bind? Specifically, are they neutralizing antibodies? Do they bind the receptor binding domain?
Well, so that's something that's ongoing. I'm assuming you mean in these convalescent samples.
Yes.
Yeah, so that's ongoing. We're going to use monoclonal antibodies with known specificities to really map out the composition of these convalescent sera. In general, we find a reasonable correlation between overall IgG ELISA titers against the spike and neutralizing potential. Based on what we know, a lot of the neutralizing antibodies do seem to be against the RBD and seem to be ACE2 blockers. And so there are ways to look at them more specifically, and that's something that we're really planning to do. But yeah, to answer your question, we haven't really done a deep dive yet to really understand. We do find this interesting phenomenon that there are discordancies between overall spike titers by ELISA and neutralizing activity using our VSV system. So you know, we don't really understand the basis of these discordancies, but a simple hypothesis is that it has to do with differences in the composition of the antibodies. So this would be something we definitely would like to try and figure out, but we haven't really gotten there yet.
Thank you.